types of mutations. Insertion, which is when a nitrogen base is inserted in the DNA code and all the other codes are shifted over. Deletion, and that's when a nitrogen base is deleted from the DNA code and all the other codons are shifted over. Insertion and deletion are also called frameshift mutations because they scoot the codons over. Another mutation is substitution, and that is when a nitrogen base is replaced with another in a DNA code.
A helpful mutation for this hen is having a variety of colors in it. This will help create diversity in its offspring.
A harmful mutation could be when a chicken has an extra foot. This will be harmful because it will have trouble walking.
This is the incubator. It is where the eggs were kept during the hatching process. It maintains a stable temperature to keep the eggs warm.
We started researching on types of chickens, DNA traits, and possible source of errors. While researching, we observed eggs' physical traits such as color of the egg and its size, and etc. The most important lessons in this project was to learn about how genes are passed from one generation to the other. By researching, we learned things like Punnett square, dominant traits, and recessive traits. Then, to finalize what we learned, we recorded our research about chicks' characteristics.
To see the development of the egg, we candle the egg. Candling is the process of using a light to see the inner contents of the egg. A light is funneled through the exit hole in a candler. This helps focus the light. Candling should be done between the 10th and 18th day of incubation. Otherwise, the light from the light source can cause biochemical changes to the development of the chick. It's important not to candle for too long. The egg can become cold and the heat radiating from the light source can cause the egg harm. These are the dominant and recessive genes for the two chickens. This is the monohybrid cross. Big B, which is dominant, is barred feathers, and little b, which is recessive, is not barred. The offspring is a heterozygous allele that is dominant barred. This is a dihybrid cross. It is homozygous rose comb and homozygous white skin times homozygous rose comb and homozygous yellow skin. When we foil it, we get big C, big S times big C, little s. Big C is rose comb, big S is white skin, and little s is yellow skin. The results for that offspring is rose comb with white skin.
After dissecting a non-hatched egg, we found that it was fertilized and had developed a blood.
Then, we faced the biggest error of the experiment. None of the eggs hatched. The possible explanation is that the eggs were laid directly after molting, which means we took the eggs away from hens too early. Even though none of the chicks hatched, we can still predict their traits based off the appearance.
Was used to compare and contrast DNA. We can see what traits belong to the parents. It's an ideal way because normal human eyes and microscopes cannot see the DNA. So we asked ourselves, how are genes passed from parent to offspring? Genes are passed through certain alleles from the parents when they reproduce. This determines unique characteristics of the offspring. In addition, to see heredity patterns, we can use gel electrophoresis.
Thanks for watching!